Right. So it is a glycolysis means so conversion of glucose into 2-pyruvic acids or pyruvates. It is common pathway for both aerobic and anaerobic organisms. It is also known as EAP pathway. It was discovered in Emden, discovered by Emden, Meerhoff, and Parnas in 1930, and it occurs in cytosol. In this, glucose is converted into 2-pyruvates, and 2 ATPs are formed as a net energy. Okay, so then, so glycolysis can also occur in anaerobic conditions or without oxygen also, right? And in aerobic conditions also. So in anaerobic conditions, the lactate is the end product of glycolysis, right? So in aerobic condition, at the end of electron transport chain, the glucose is completely oxidized into H2O and CO2. So yeah, glycolysis is major pathway for ATP production in tissues lacking mitochondria like erythrocytes, cornea, and lens cells. Right. Okay. So glycolysis is a central metabolic pathway. So why? With many of its intermediates providing branch point to other pathways. Okay, what are the intermediary substances which are formed in the glycolysis? They are linked to other pathways also. So it acts as a central pathway. Okay, so the intermediates of glycolysis are useful for synthesis of amino acids and the fat. Okay, so that is in that way also it is central pathway. Right. So here reactions of glycolysis. It can be divided into 3 distinct phases. So first one is energy investment phase or priming phase. Second one is it is single reaction, splitting phase, and the third one is energy generation phase. So in first phase, the energy is consumed, you remember. Okay, in third phase or last phase, the energy is produced. Okay, right. So in first reaction of the glycolysis, glucose, which is obtained from the digestion of foodstuffs, okay, like lactose, sucrose, maltose, glycogen, starch, okay, is brought into the hexose phosphate pool. Okay, not hexose phosphate pool through the action of enzyme hexokinase. Okay, that means glucose is converted into glucose 1-phosphate or 6-phosphate. Okay, so in that way, it that means in the cell there are many hexose phosphates are present. Okay, so and a newly formed glucose is also added to that pool. Right. Right. So this is the first reaction, that is phosphorylation. Right. So here the free glucose is phosphorylated to glucose 6-phosphate by hexokinase. You remember this enzyme? It's very, very crucial in the glycolysis. Okay, we will come again in the regulation part. Right. So here one ATP is consumed. Okay, so ADP is produced and Mg+2 ions are they act as cofactors for hexokinase. So hexokinase is common for all the tissues, but glucokinase is specifically meant for the liver cells. And here hexokinase means it can act on fructose, it can act on galactose. are other isomers of glucose, but glucokinase is specially act on only glucose and it is present in the liver. Okay, that's it. So glucose 6-phosphate is formed. That means glucose is phosphorylated. Okay, so exokinase splits the ATP into ADP and inorganic phosphate. The inorganic phosphate is added to the glucose. The separated inorganic phosphate is added to the glucose to form glucose 6-phosphate. And I have already told you, exokinase is a key glycolytic enzyme. Okay, so this will come in the regulation part, right? So isomerization. See, you should know that glucose is a 6-carbon compound. Okay, so and it is aldohexose. It is an aldohexose. And in the second step, the glucose is glucose 6-phosphate is isomerized. That means from aldose form to ketose form, it is converted. Okay, so here glucose 6-phosphate is a central molecule with a variety of metabolic fates, glycolysis, glycogenesis, gluconeogenesis, and HMP shunt. Okay, the isomerization of glucose 6-phosphate, which is an aldose sugar, to fructose 6-phosphate, a keto sugar, is catalyzed by phosphohexose isomerase. You remember, most of the glycolytic reactions are reversible reactions. Okay, so the fructose 6-phosphate can also generate glucose 6-phosphate if at all necessary, particularly in gluconeogenesis. Anyway, the enzyme is phospho. Okay, because here phospho is there. Okay, phosphate is there, and hexose because these two molecules are 6-carbon compounds and isomerase. So glucose is the isomerase of fructose. Fructose is the isomer of glucose. So that's why phosphohexose isomerase. Here also magnesium ions are assisted. Okay, that is isomerization. This is first isomerization reaction in the glycolysis, right? So then, in the first reaction, the phosphorylation we have seen. So this is another reaction where again phosphorylation occurs. Here you can see fructose 6-phosphate, which is formed earlier, is converted into fructose 1,6 biphosphate. Okay, so actually, manaku structures idea unte idi clear about. So fructose is a 6-carbon compound. So fructose 6-phosphate means there is a phosphate at 6th carbon, and here again the ATP is giving one more phosphate group. Okay, that is attached to first carbon. So to form fructose 1,6 bisphosphate or biphosphate. Here you see in the fructose 1,6 phosphate, there is a phosphate at first carbon, there is a phosphate at 6th carbon. Okay, so that's why it is known as fructose 1,6 biphosphate, and this reaction is very, very regulating, very, very important. See, PFK, you remember this enzyme. So, ఇది జేఎల్లో అడగారు, నెట్ లాంటి సెట్ లాంటి ఎగ్జామ్స్ లో అడుగుతారు. So it is very, very regulating, important regulating enzyme. The PFK reaction is a rate limiting step, which is a very crucial step in the glycolysis. And this third one, you remember, third step is very, very rate limiting step. Okay, if at all the glycolysis to be stopped, so this is the most common and commonly stopped step by inactivating this PFK enzyme. Anyway, let's see that later. It is controlled by concentration of substrates C, ATP and fructose 6-phosphate. Okay, so here whenever cell contains sufficient amount of ATPs, so there is no need of glycolytic pathway. There is no need of glucose oxidation. So the excess amount of ATP binds to this PFK and inactivates it. Or in the same way, fructose 6-phosphate, the substrate here. Okay, it goes and binds with TFK so that the reaction is stopped. Okay, all the fructose 6-phosphate are accumulated. So glycolysis is not proceeded. So this is a rate limiting or regulating a reaction in the glycolytic pathway. Okay, you should remember third reaction, PFK, also very, very important for the regulation of the glycolysis. And splitting, you should remember here one thing is fructose is a 6-carbon containing enzyme. And here in the previous reaction also, you see, it is irreversible reaction. In that way also, it is important. There are 3 irreversible reactions, 1, 3, and 10. 1, 3, and 10. You remember. Okay. So, anyway, here in this reaction, fructose 1,6 bisphosphate, which is 6-carbon containing, the compound, it is splitted, cleaved, okay, dissociated. Okay, 2 compounds. One is glyceraldehyde 3-phosphate. Another one is dihydroxyacetone phosphate. Both are 3-carbon containing carbohydrate molecules, simplest carbohydrate molecules. Okay, with a phosphate. Okay, glyceraldehyde and dihydroxyacetone are the, they are the isomers, okay, to each other. One is aldotriose, another one is ketotriose. Okay, here it contains aldehyde functional group, here it contains keto functional group. Anyway, this one contains 3 carbons and this one contains 3 carbons. So that's why it is famous enzyme. It also comes in other pathways, aldolase. So it breaks a 6-carbon containing compound into 2 3 carbons containing compounds. Okay, one is glyceraldehyde, another one is dihydroxyacetone phosphate. Okay, so this is also very, very important, and we should not be confused. This is 4th reaction. Why? See, the dihydroxyacetone phosphate, which is formed here in addition to glyceraldehyde 3-phosphate. Okay, it is isomerized. See, this is second isomerization reaction. 2 phosphorylation we have seen and 2 isomerations. Okay, this is second isomerization reaction. So, isomerization of dihydroxyacetone phosphate. So, this 3-carbon containing dihydroxyacetone phosphate, which is a triose, ketotriose, containing 3 carbons. Okay, so see, this is not phosphohexose isomerase. This is triose phosphate isomerase enzyme. Triose phosphate isomerase enzyme. So it's a mistake. I'll change it in the notes. Anyway, so the enzyme triose phosphate isomerase enzyme converts dihydroxyacetone phosphate into another glyceraldehyde 3-phosphate. We should remember that. So now from a single glucose, we have 2 glyceraldehyde 3-phosphates. Okay, now we have 2 glyceraldehyde 3-phosphates. So from now on, if we are discussing a single reaction, that means 2 reactions will be occurred. Okay, ఇక్కడ నుంచి ఒక reaction is equal to 2 reactions which are similar, same reaction. So ఇప్పుడు మనకు 2 glyceraldehyde 3-phosphates ఉన్నాయి, right? So here see, next reaction is oxidation. So oxidation of what? Oxidation of glyceraldehyde 3-phosphate. So ఇక్కడ ఒక రియాక్షన్ జరుగుతుందంటే సేమ్ రియాక్షన్ ఇంకోటి కూడా జరుగుతుంది. ఎందుకంటే మనకు 2 గ్లిజరాల్డిహైడ్ 3 ఫాస్ఫేట్స్ ఉన్నాయి. ఇక్కడ నుంచి ఏది ఫామ్ అయినా అది 2 ఉన్నట్టుగా మీరు గుర్తుంచుకోవాలి ఫ్రమ్ ది 6th రియాక్షన్. ఓకే, సో గ్లిజరాల్డిహైడ్ 3 ఫాస్ఫేట్ is converted into 1,3 bisphospho or biphospho glycerate, which is also a triose. Okay, so here 2 electrons are lost from the glyceraldehyde 3 phosphate and 2 hydrogens are also lost. Okay, that is transferred to NAD plus to form. Okay, so NADH plus H plus. See, let's see. This step is important as it is involved in the formation of NADH2. ఎన్నో రియాక్షన్ లో NADH2 ఫామ్ అవుతుంది. 6th రియాక్షన్ లో NADH2 ఫామ్ అవుతుంది. ఎన్నో రియాక్షన్ లో ATPs యూటిలైజ్ అవుతుంది. ఫస్ట్ రియాక్షన్ అండ్ థర్డ్ రియాక్షన్ లో ATP యూటిలైజ్ అవుతుంది, గుర్తుంచుకోవాలి, రైట్? సో ఆల్రెడీ 2 ATPs are utilized and here in the 6th reaction NADH2 is produced. Okay, so this NADH2 is converted into ATP in electron transport chain. Okay, so here this step is important as it is involved in the formation of NADH2 and a high energy compound. So this is 1,3 bisphosphoglycerate. Why? It contains 3 phosphates. So that's why that is considered as a high energy compound. So from which 2 ATPs are produced. Right. In aerobic condition, NADH passes through electron transport chain and 6 ATPs are synthesized by oxidative phosphorylation. ఇది మనం గుర్తుంచుకోవాల్సిన అవసరం లేదు. ఓకే, సో జస్ట్ గ్లైకాలసిస్ వరకు ఇది అవసరం లేదు. ఓకే, అది ఎలక్ట్రాన్ ట్రాన్స్పోర్ట్ సిస్టమ్ లో దట్ ఈస్ యూటిలైజ్డ్ విత్ దిస్ NADH2. ఎనీవే, సో హియర్ గ్లిజరాల్డిహైడ్ 3 phosphate is converted to 1,3 bisphosphoglycerate గా కన్వర్ట్ అవుతుంది. అండ్ iodoacetate, arsenate, they are the inhibitors of this reaction. Okay, they block this enzyme. Anyway, it is not asked in the examinations. Net gaani set gaani adutaru andulo, right? See, next reaction is in the 6th reaction lo NADH2 form ayindi. 7th reaction lo ATP form avutundi, right? So here 7th reaction is generally known as substrate level phosphorylation. So generally ATP is synthesized with the help of ATP synthase and oxidative phosphorylation takes place. So then only the ATP is produced from the ADP. But here one of the phosphates possessed by this 1,3 biphosphoglycerate is converted into first of all, the one phosphate from the first carbon is removed and it is transferred to ADP to produce ATP. So here the ATP is produced not by the enzyme but by the substrate. Okay, so with the help of Mg+2 ions. So this is also a reversible reaction. And here wherever you see kinase, there is an involvement of phosphate, either it is removed or added. So here one phosphate is removed from the first carbon of the 1,3 bisphosphoglycerate to 3 phosphoglycerate, right? So that is 7th reaction. Here what happens, how the ATP is produced? The step is just substrate level phosphorylation. The formation of ATP by phosphate group transfer from a substrate such as the 1,3 bisphosphoglycerate is referred to as a substrate level phosphorylation. Okay, ఇందులో ఏం జరుగుతుంది, ఒక సబ్స్ట్రేట్ నుంచి ఫాస్ఫేట్ అనేది సో ట్రాన్స్ఫర్ అనేది చూస్తాం, అందుకే దాన్ని సబ్స్ట్రేట్ లెవెల్ ఫాస్ఫోరిలేషన్ గా మనం జనరల్ గా చెప్పుకుంటాం, రైట్? సో అన్లైక్ మోస్ట్ అదర్ కైనేస్, దిస్ రియాక్షన్ ఈస్ రివర్సిబుల్. మోస్ట్ ఆఫ్ ది అదర్ కైనేస్ రియాక్షన్స్ ఏంటంటే ఫాస్ఫేట్ ను రిమూవ్ చేసినప్పుడు లేదా ఫాస్ఫేట్ యాడ్ చేసినప్పుడు ఏమవుతుంది, మోస్ట్లీ దే ఆర్ irreversible reactions. But here this is also a reversible reaction. We have now 3-phosphoglyceric acid or 3-phosphoglycerate. with a generation of ATP. ATP seventh reaction low first ATP form అయింది గుర్తుంచుకోవాలి. Right. So then mutation reaction. Okay. So the 3-phosphoglycerate is converted into 2-phosphoglycerate by phosphoglycerate mutase. So name itself is explaining. So there will be a change in the structure. Okay. So phosphoglycerate 3-phosphoglyceric acid or 3-phosphoglycerate is converted into 2-phosphoglycerate. That means the phosphate group from third carbon to second carbon, it is transferred. This is also an isomerization reaction. Okay, so both are same compounds. Okay, but the phosphate group is here, at third carbon. Here it is at second carbon. So the enzyme phosphoglycerate mutase catalyze this reaction. And then a small reaction, that is dehydration, takes place. Here, the high energy compound phosphoenol pyruvate, this one, is generated from 2-phosphoglycerate. Okay, so 2-phosphoglycerate నుంచి PEP అనేటువంటి ఒక substrate product అనేది ఫామ్ అవుతుంది. So this reaction is catalyzed by the enzyme enolase. Okay, so this enzyme also requires Mg+2 or Mn+2 and enolase is inhibited by fluoride ions. Fluoride ions inhibits the enolase enzyme. Okay, so anyway, PEP, phosphoenol pyruvate, is formed from the 2-phospho. This is 2-phosphoglycerate, right? So spelling mistake on 2-phosphoglycerate. Manchi phosphoenol pyruvate అంటే ఒక డబుల్ బాండ్ అనేది ఫామ్ అవుతుంది డ్యూ టు లాస్ ఆఫ్ H2O from the 2-phosphoglycerate. Phospho N ol. Okay, so at second carbon there will be a double bond formed between the first and second carbons. Okay, so and phosphate contain. So this is called phosphoenol pyruvate, PEP, the famous substrate, right? Just let me take some water. Okay. Now, after formation of phosphoenol pyruvate, which is high energy substance, okay, so with the enzyme enolase and Mg+2 ions are Mn+2, they act as cofactors. So phosphoenol pyruvate is a high energy compound and unstable, right? The enzyme pyruvate kinase, see 10th reaction, irreversible reaction, and pyruvate kinase enzyme is also rate limiting enzyme. Okay, anyway, here we have 2 isomerization reactions, we have 2 phosphorylation reactions, and we have 2 substrate level phosphorylation reactions. Okay, so here the enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphoenol pyruvate to pyruvic acid. Here the phosphate which is present in the PEP is transferred to ATP. So from a substrate to ADP to form ATP. So that's why this is also considered as a substrate level phosphorylation. Okay, so here the pyruvate is formed in aerobic reaction according to aerobic reactions or aerobic respiration, cellular respiration. So with this reaction, the glycolysis is over. Okay, anyway, here you see. So these are the reactions: glucose 6-phosphate, fructose 6-phosphate isomerization, fructose 1,6 bisphosphate. Okay, so splitting stage where DHAP and the glyceraldehyde 3-phosphate is formed. And this DHAP is converted into glycerol 3-phosphate in the fifth reaction. In the sixth reaction, NADH2 is produced from glycerol 3-phosphate. So 1,3 biphosphoglycerate is formed, and in the seventh reaction, first ATP is produced, okay, by kinase enzyme. Okay, to form one phosphate is removed, 3-phosphoglyceric acid formed. 3-phosphoglyceric acid is converted into 2-phosphoglycerate, which is catalyzed by an enzyme called mutase. And then 2-phosphoglycerate is converted into phosphoenol pyruvate, which is high energy compound. Okay, PEP. Right. So then PEP is converted into pyruvate. Okay. In aerobic respiration, you remember the pyruvate is converted into acetyl-CoA, which enters into Krebs cycle in aerobic respiration. But in anaerobic conditions, okay, like exercising muscle, RBCs, okay, in such conditions, the pyruvate is converted into in microorganisms like anaerobic microorganisms. The pyruvate is converted into lactic acid with the help of enzyme lactate dehydrogenase. And here, the free NADH is converted into NAD is converted to NADH H2 H plus, right? So this is lactate fermentation, right? Anyway, in anaerobic condition, the lactate formation is last step. In aerobic condition, pyruvate formation is last step. Okay. Regulation of glycolysis. ఇలాంటి క్వశ్చన్స్ సెట్ లాంటి ఎగ్జామ్స్ లో నెట్ లాంటి ఎగ్జామ్స్ లో అడుగుతారమ్మా. రెగ్యులేషన్ అనేది యాక్చువల్గా మనకు లేని లేదు, బట్ స్టిల్ యూ షుడ్ హ్యావ్ సమ్ బేసిక్ నాలెడ్జ్ అబౌట్ ద రెగ్యులేషన్ ఆఫ్ glycolysis, how it is regulated. So there are 3 regulatory enzymes. One is hexokinase, okay, and phosphofructose kinase PFK, and pyruvate kinase. Okay, these 3 enzymes are rate limiting or regulating enzymes of glycolysis. So they catalyze the irreversible reactions to regulate glycolysis. Okay, so that is about the the regulatory enzymes of food. Okay, now hexokinase. So this is first enzyme. Okay, hexokinase it converts a glucose into glucose 6-phosphate. Okay. Hexokinase is inhibited by glucose 6-phosphate, which is its product. Okay, మనకు ATP అవసరం లేనప్పుడు, so this is called product inhibition. సో హెక్సోకైనేజ్ యొక్క ప్రోడక్ట్ ఏంటి? గ్లూకోజ్ కన్వర్షన్ ఆఫ్ గ్లూకోజ్ టు గ్లూకోజ్ 6-phosphate. మనకు కావాల్సినన్ని ఏటీపీలు ఉన్నావు కాబట్టి, సో అప్పుడు గ్లూకోజ్ 6-phosphate అనేది డైరెక్ట్ గా వెళ్ళి ఇట్ యాక్ట్స్ on hexokinase enzyme, which is an allosteric compound, the enzyme. So it will become inactive due to the action of glucose 6-phosphate. This enzyme prevents the accumulation of glucose phosphate due to product inhibition. Okay, word sentence forms like that. Okay, glucose 6-phosphate and okay, so this point is not appropriate. Anyway, so glucokinase enzyme is the hexokinase of liver. Glucokinase, which specifically phosphorylates glucose, is an inducible enzyme. So ఇక్కడ మనకు 2 రకాల ఎంజైమ్స్ పనిచేస్తే, ఒకటి glucokinase, another one is hexokinase. Glucokinase is present in the liver. Okay, that should work very continuously, then compare with the other cells. Okay, so the substrate glucose probably through the involvement of insulin induces the glucokinase. Okay, so anyway, hexokinase is regulated by glucose 6 phosphate. So product inhibition. Then wala glycolysis అనేది regulate చేయబడుతుంది లేదా ఆపబడుతుంది. So PFK, phosphofructokinase, is the most important regulatory enzymes in glycolysis. Okay, so then PFK is an allosteric enzyme regulated by allosteric effectors. ఇది గుర్తుంచుకోవాలమ్మా. ATP, citric acid, and H+ ions. ATP and citrate or low pH are the most important allosteric inhibitors. Okay, ultimately, respiratory enzyme యొక్క aim ఏంటి అంటే production of energy, right? So the produced ATP or citric acid, which is formed in the first reaction of the Krebs cycle, and H+ ions, which are released many places. Okay, so all these three are, they act as inhibitors. If at all the cell contains high amount of ATP, these three kind of substance, okay, so they go and bind to allosteric region of phosphofructokinase. Okay, allosteric means other than the active site. Active site కాకుండా సో వేరొక allosteric ఏరియాలో వెళ్ళి బైండ్ అయ్యి పిఎఫ్కేను అవి స్విచ్ ఆఫ్ చేస్తాయి అన్నమాట. ఓకే, ఇన్యాక్టివ్ చేస్తాయి. ఇన్ ద సేమ్ వే fructose 2,6 biphosphate, ADP, AMP, and inorganic phosphate are the allosteric activators. So if we, whenever either of these bound to the enzyme PFK, okay, so then also the the reaction, okay, is stopped. So PFK is stopped. Okay, phosphofructokinase, sixth reaction, I think. 1, 2, 3, 4, 5, 6, 7th reaction. Sorry, third one. Okay, so 1, 3, 10, right? Third reaction is very, very crucial. Phosphofructokinase contains an allosteric site, so that some of the compounds, they may be from the same pathway or from outside, they come and bind to this phosphofructokinase, okay, and change its conformational form, or conformational form is changed by allosteric activators or inhibitors. Okay, so these were ATP, citric acid, H+ ions are they are inhibitors of PFK. Fructose 2,6 biphosphate, ADP, AMP, and inorganic phosphates they are the activators. Okay, so you should remember the compounds that activate the PFK and inactivate the PFK, right? ATP, citrate, and H+ ions, they are the inhibitors. ADP, AMP, fructose 2,6 bisphosphate are the allosteric activators, right? It also extra, right? So third enzyme is pyruvate kinase. Kinase means so either it adds phosphate or removes phosphate. Okay, so pyruvate kinase inhibited by ATP, okay, so and activated by fructose 1,6 biphosphate. Okay, so pyruvate kinase is active in dephosphorylated state and inactive in phosphorylated state. Okay, so here PK is inhibited by end product like ATP, okay, and activated by fructose 1,6 biphosphate అనేది. So general ga it activates the pyruvate kinase. Okay, so pyruvate kinase is active in dephosphorylated state and inactive in phosphorylated state. It is opposite to general reactions. ఏదైనా ఒక సబ్స్టెన్స్ కి ఫాస్ఫేట్ అందిస్తే that will be activated, but here the reverse is happened. Okay, so inactivation of pyruvate kinase is brought about by cyclic AMP dependent protein kinase. The hormone glucagon inhibits hepatic glycolysis by this mechanism. Right, what you should remember is three enzymes. One is hexokinase, another one is phosphofructokinase, another one is last enzyme pyruvate kinase. Okay, so these three enzymes are rate limiting enzymes. Okay, so if at all the glycolysis to be regulated, so various activators or deactivators, okay, they bind to those enzymes and they either stop them or they may activate according to the cell need, cell economy. Okay, so that is about the regulation of glycolysis. And here these are important points we should remember for the JL examination. Right, so energy yield from glycolysis. Okay, so here the glycolysis is common for both aerobic and anaerobic organisms. So during anaerobic respiration, one molecule of glucose is converted into 2 molecules of lactate. So I told you that. So here the pyruvate is formed, means 2 pyruvates are formed because 2 glycerol head 3 phosphates are produced, and pyruvate is 3 carbon compound, right? So glucose is 6 carbon compound. So in that way, simply a 6 carbon compound is split into 2 3 carbons containing compound pyruvates. So from 2 pyruvates, 2 lactates are produced in anaerobic respiration. That is one point. Okay, and then yeah, so a net yield of 2 molecules of ATP. Okay, what about the NADH2? Okay, so that NADH2 is converted into here pyruvate, another NADH2 is produced. Okay, this reaction should be, yeah, here the NADH2 is utilized. Okay, ఇది కూడా mistake ఉందమ్మా. So during the formation of lactate from the pyruvate, the NADH2 is utilized, the NAD is produced, and this NAD is again here that is utilized. Okay, pyruvate nundi lactate form అయ్యే రియాక్షన్ లో NADH2 ఇట్లా చేంజ్ చేద్దాం. సరే, నేను నోట్స్ ఇచ్చేటప్పుడు చేంజ్ చేస్తాను. ఇక్కడ ఏమవుతుందంటే NADH2 is utilized and NAD plus is formed, and this NAD plus is utilized in this 6th reaction. Okay, so that is how in anaerobic respiration the NAD is reused, and then yeah, so net మనం ఇక్కడ నెట్ ఏం చెప్తాం, just 2 molecules of ATP in the anaerobic glycolysis. Okay, so 4 molecules of ATP are synthesized by 2 substrate level phosphorylation. 2 ATP molecules are used in step 1 and 3. Okay, it is both for aerobic and anaerobic. Okay, step 1 and 3 లో మనం 2 ATP utilization జరుగుతుంది కాబట్టి, so net yield in anaerobic glycolysis is 2 ATPs, right? So during aerobic condition, okay, ఇక్కడ చూడండి, dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. ఆల్రెడీ మనకి ఇక్కడ ఒకటి ఉంది glyceraldehyde 3-phosphate, ఇక్కడ ఇంకోటి ఫామ్ అయింది దీని నుంచి. కాబట్టి ఒక గ్లిజరాల్డిహైడ్ 3-phosphate కి ఒక NADH2 ఏర్పడుతుంది. రెండు గ్లిజరాల్డిహైడ్స్ ఉంటాయి కాబట్టి రెండు NADH2లు ఏర్పడతాయి. సింపుల్. ఓకే, సో ఆ రకంగా ఇన్ దట్ వే 2 NADH2 molecules are generated in the glyceraldehyde 3-phosphate dehydrogenase reaction and enter electron transport chain. Okay, so ఈ NADH2 అనేది electron transport chain లో it is converted into ATP. ఓకే, ఇక్కడ ఏమని ఇచ్చాడు చూడండి, NADH provides 3 ATP. ఒక్క సింగిల్ NADH provides 3 ATPs. సో ఈ రకంగా రెండు NADH2 ఉన్నాయి కాబట్టి సో 6 ATPs are produced. Okay, so total ATP net ఏమో 2 ATPలు అండ్ ఇక్కడ NADH2 నుంచి కూడా మనం యాడ్ చేసుకుంటే మొత్తం 8 ATPs అనేటువంటి ఓవరాల్ రెస్పిరేషన్ లో గ్లైకాలిసిస్ పార్ట్ ఎంత అంటే 8 ATPs. In glycolysis 2 net ATPs are produced. Each ATP formation requires, చూసుకోండి, ఒక్క ATP ఫార్మేషన్ కి ఈ పాత్వేలో 12 kilocalories or 50 kilojoules of joules per mole energy is required. So one glucose possesses 686, ఇది వేరే పాయింట్, 686 kilocalories of energy, or ఇది గుర్తుంచుకున్నాము, ఒక ఎగ్జామ్ లో అడిగిండు, జౌల్స్ లో అడిగేసరికి నేను కన్ఫ్యూజ్ అయినా రాంగ్ ఆన్సర్ పెట్టిన. రైట్, సో 2870 కిలో జౌల్స్ ఆఫ్ ఎనర్జీ అందులో యాజ్ ఎ కెమికల్ ఎనర్జీగా ఉంటుంది అందులో ఒక గ్లూకోజ్ మాలిక్యూల్. సో అవుట్ ఆఫ్ దిస్, సో ఓన్లీ 3% ఎనర్జీ అంటే 686 కిలో క్యాలరీస్, ఓన్లీ 3% ఎనర్జీ ఈస్ కన్వర్టెడ్ ఇంటూ ATP. ఇందులో గ్లైకాలిసిస్ లో, okay, glycolysis only 3% of energy, that is so 24 kilocalories of energy from 686 kilocalories of energy, that is 3% of energy is converted into ATP, right? So manaku aerobic respiration, aerobic glycolysis lo rendu net ATPలు అనేటువంటి ఫామ్ అవుతాయి. నెంబర్ 1, 2 NADH2లు ఫామ్ అవుతాయి కాబట్టి ఒక్కొక్క NADH నుంచి 3 ATPs ప్రొడ్యూస్ అవుతాయి. ఇక్కడ ఎలక్ట్రాన్ ట్రాన్స్పోర్ట్ చైన్లో, ఇక్కడ అంటే ఓవరాల్ గా చూసుకుంటే ఒక గ్లూకోజ్ నుంచి గ్లైకాలిసిస్ కంట్రిబ్యూషన్ ఎంత, 38 ATPs అనుకుంటే లేదా 36 ATPs అనుకుంటే అందులో 8 ATPs ఆర్ ఫ్రమ్ గ్లైకాలిసిస్. అది గుర్తుంచుకోవాలి. దట్ ఈస్ ద ఎనర్జెటిక్స్ ఆఫ్ glycolysis. And next here, so how the pyruvate is converted into lactate, ఇందాక మనం ఆల్రెడీ చూసినాం. చూడండి ఇక్కడ ఇది కరెక్ట్ రియాక్షన్. NADH2 is utilized anywhere. Here this is pyruvic acid, this is lactic acid, this is pyruvate, this is lactate. ఇక్కడ H ఉంటే యాసిడ్ అంటాం, లేకపోతే 8 అంటాం, సింపుల్ అంతే, రైట్. ఇన్ ఎనరోబిక్ కండిషన్ pyruvate is reduced, okay, to lactate by lactate dehydrogenase, LDH is also 5-isoinosine. I have to move it. The cardiac isozyme LDH will be increasing myocardial infarcts. ఇది ఒక స్టెప్ చూడండి ఏమవుతుంది ఇక్కడ అంటే, దిస్ ఈస్ పైరువేట్ with the help of enzyme lactate dehydrogenase, it is converted into lactic acid, CH3CHOH COO minus. Okay, so here it is reduced and hydrogens are added, okay, to this pyruvate. Okay, so here NADH2, which is formed in the previous reaction, it is utilized here and again NAD plus is ready. మళ్ళీ ఇది పోయి 6th reaction లో అక్కడ NADH2 formation help అవుతుంది, right? So this NAD regenerated is again used in will be used in the earlier reactions of glycolysis. So this is lactic acid, right? So then overall lactic acid reaction is this one: glucose plus 2 ADP plus 2 inorganic phosphate, that gives 2 lactates and 2 ATPs and 2 H2O, 2 H plus ions out, right? And then yeah, ఇందాక నేను చెప్పి ఇది మళ్ళీ ఎర్లియర్ రియాక్షన్స్ లో యూటిలైజ్ అవుతుంది. సేమ్ గ్లిజరాల్ హెడ్ 3 ఫాస్ఫేట్ 1,3 బయోఫాస్ఫో గ్లిజరేట్ అనేది ఫామ్ అవుతుంది. దాని నుంచి ఇక్కడ గ్లిజరాల్ హెడ్ 3 ఫాస్ఫేట్ నుంచి 1,3 బిస్ఫాస్ఫో గ్లిజరేట్ ఫామ్ అయ్యే దగ్గర NADH2 అనేది ఫామ్ అవుతుంది. ఆ NADH ఇక్కడ యూటిలైజ్ అవుతుంది. దేనికి? కన్వర్షన్ ఆఫ్ పైరువేట్ టు లాక్టేట్. సో దానివల్ల ఏం ప్రొడ్యూస్ అవుతుంది? NAD ప్లస్ ఫామ్ అవుతుంది. దిస్ NAD ప్లస్ ఫామ్ అగైన్ ఇట్ ఈస్ యూటిలైజ్డ్ ఇన్ ద సేమ్ రియాక్షన్. ఓకే, సో గ్లిజరాల్ హెడ్ 3 ఫాస్ఫేట్ టు 1,3 బిస్ఫాస్ఫో గ్లిజరేట్ లో మళ్ళీ NAD ప్లస్ అనేది ఫామ్ అవుతుంది. సో పైరువేట్ అనేది ఎస్టైల్ కోయాగా మారదు, ఎందుకంటే ఆక్సిజన్ అనేది ఉండకపోవడం వల్ల, రైట్? సో దట్ ఈస్ యునో glycolysis. And then alcohol fermentation, ఇది కూడా ఒకటే రియాక్షన్, బట్ 2 steps ఉంటాయి. Yeast and some microbes which are anaerobes. Okay, so they by fermentation they convert glucose into ethyl alcohol. మనకు ప్రొడ్యూస్ అయ్యే కమర్షియల్లీ ప్రొడ్యూస్ అయ్యేటువంటి ethyl alcohol రియాక్షన్ ఇది అన్నమాట. They are anaerobes, they produce ethyl alcohol. Okay, so glucose is converted into ethyl alcohol and CO2. It occurs in 2 steps. First, pyruvate is converted into acetaldehyde. It is very, very harmful by pyruvate decarboxylase. Okay, so here thiamine pyrophosphate TPP Mg plus 2, they act as coenzyme cofactors, and here first of all decarboxylation takes place with the help of enzyme pyruvate decarboxylase to form acetaldehyde, which is very, very harmful. Immediately it is harmful to human being. Okay, anyway, it is a toxic compound. Anyway, this acetaldehyde, so now we are discussing it, yeast and some alcohol microbes, right? So this acetaldehyde immediately reduced, okay, so into ethanol CH3CH2OH or ethyl alcohol. సో మన కమర్షియల్ గా ఇథైల్ ఆల్కహాల్ ఇలాగే ప్రొడ్యూస్ చేస్తారు. ఓకే, సో ఎనీవే దిస్ ఇస్ హౌ ద alcohol fermenter bacteria or microbes like yeast, okay, so they convert pyruvic acid at the end of the glycolytic pathway by 2 enzymes, pyruvate decarboxylase and alcohol dehydrogenase. So this alcohol dehydrogenase converts acetaldehyde into this what we call the
ethanol.
Yeah, ethanol or ethyl alcohol. Okay, so with this the glycolysis is completed. Okay, here same thing happens. NADH2 is consumed, and that is reutilized in the 6th step. Okay, so then I'll just introduce this Krebs cycle. I'll just introduce. So Krebs cycle, I'll explain this reaction. Right here, the glycolysis that occurs in cytosol or cytoplasm. Okay, but the next pathways they are occurred in the mitochondria. Okay, so for that purpose the pyruvate should be converted into acetyl coenzyme A. So because pyruvate is not readily taken into the mitochondria. It is impermeable to pyruvate. Okay, so for that purpose, first of all, it is decarboxylated. The pyruvate is decarboxylated. Then only the mitochondria membrane allows the pyruvate. Okay, so anyway, the Krebs cycle was discovered in 1930s by Sir Hans Krebs. Okay, here the energy of glucose is still there in the pyruvate. Okay, so it should be more oxidized. Okay, so the pyruvate enters into Krebs cycle. So where it occurs in matrix of mitochondria. Okay, so this there is pyruvate H plus importer. Okay, so which allows an decarboxylized pyruvate. Okay, it is a carrier protein that allows decarboxylated pyruvate into the mitochondrial matrix. Okay, so ultimately the acetyl coenzyme A produced, and it is regarded as the connecting link molecule between the glycolysis and Krebs cycle. Okay, so yeah, acetyl coenzyme is formed like this. So at the end of the glycolysis, we have two pyruvate molecules. We have pyruvates, right? CH3, C11O, COO minus. Okay, which is pyruvate, and this is mitochondria. Here it cannot directly enter into the mitochondria, so that's why first of all this one, this is the COO minus functional group or MIT. It is decarboxylated. Okay, this CO2 is removed, right? So it is removed oxidatively. So by the enzyme decarboxylase, pyruvate decarboxylase. So, sorry, sir. The multi-complex enzyme pyruvate dehydrogenase, it catalyzes the three steps of the this conversion of pyruvate into acetyl-CoA. Anyway, first decarboxylation takes place. Okay, so that enters an acetate is formed, CH3COO minus, which is known as acetate. Okay, so then in the second step, the acetate is reduced, and a coenzyme A, which is a byproduct of vitamin B complex. Okay, so sulfur containing compound, which is known as coenzyme A, is added to that acetate, so to form ultimately acetyl coenzyme A. So in this way, the coenzyme A, slide was same. Anyway, ultimately the acetyl coenzyme A is formed in the mitochondrial matrix. It is readily and easily it can enter into the next pathway, that is the citric acid cycle. We can also call it as tricarboxylic acid cycle. Okay, Krebs cycle. So here you can see the first compound in the citric acid cycle is formed is the citric acid. That's why this is known as Krebs cycle is also known as citrate. Anyway, in the first step of the citric acid cycle, the oxaloacetate, which is the end product of citric acid, is combined with this acetyl coenzyme A, which is formed earlier. Okay, to form citrate or citric acid. So that's why as first compound to be formed is citric acid. This compound, this cycle is also known as citric acid cycle number 1. As it was discovered by Krebs, Sir Hans Krebs, it is also known as Krebs cycle. And here 1, 2, 3, 3 carboxyl groups. 1, 2, 3, 3 carboxyl groups. 1, 2, 3, 3 carboxyl groups. So the compounds which are formed in the first 3 steps of the the citric acid cycle or Krebs cycle, they are tricarboxylic acids. Okay, tricarboxylic compounds or acids. Okay, so like citrate, econitate, and isocitrate. These 3 are the tricarboxylic compounds. So that's why this pathway is also known as, TCA cycle, tricarboxylic acid cycle. Okay, so then just I'll give some, just I'll read this cycle. Just listen, right? So in the first step, the oxaloacetate, which is formed at the end of the citric acid cycle, it combines with the acetyl coenzyme A in the presence of citric acid synthase and the water molecule. Okay, the CoAsh or coenzyme A is removed from the the acetyl CoA. Okay, and this moiety is added here. Okay, CH2CO acetate is added to form citrate or citric acid. Okay, and then here the H2O is added, that is hydration, and here H2O is removed, that is the dehydration. Okay, reaction. So the enzyme econitase catalyzes this reaction, so that converts citrate to cis econitate, and then same enzyme converts it into econitate. Okay, so CO minus atom. So anyway, econitic acid is formed. Okay, ఇక్కడ 2 reactions జరుగుతాయి, ఐసోమరేషన్ కూడా జరుగుతుంది. సిస్ ఎకోనిటేట్ నుంచి సో ఎకోనిటేట్ అనేది ఫామ్ అవుతుంది. ఎనీవే ఇక్కడ అది ఇవ్వలేదు. సేమ్ ఎంజైమ్. Okay, so again by addition of an H2O molecule, okay, it rearranges it into isocitrate. The difference is ఇక్కడ double bond ఉంది, the double bond అనేది remove చేయబడుతుంది. So this is called isocitric acid. Okay, so now first carbon dioxide is removed in the respiration is during conversion of pyruvic acid into acetyl CoA. అక్కడ ఒక CO2 ఫస్ట్ ఫామ్ అవుతుంది, ఇక్కడ సెకండ్ CO2 అన్నమాట. Okay, so CO2 is formed here, where the isocitrate is converted into alpha ketoglutarate. So here oxidative decarboxylation occurs, and the hydrogens are also removed here. Isocitrate dehydrogenase enzyme catalyzes this reaction to form alpha ketoglutarate. Okay, alpha ketogluteric acid, and then alpha ketogluteric acid by the enzyme alpha ketoglutarate dehydrogenase. Again here dehydrogenase enzyme is seen. Okay, so here one coenzyme A is added, which is high energy compound, to this alpha ketogluteric acid. Okay, in the presence of alpha ketoglutarate dehydrogenase complex enzyme. So that with that, this alpha ketoglutarate is converted into succinyl CoA. Okay, succinyl coenzyme A. Now it is high energy compound. Okay, so here a substrate level phosphorylation takes place from a substrate during the reaction. The energy which is released in a reaction, if it is utilized for the formation of a triphosphate a nucleotide like ATP or GTP, so that reactions are generally considered as or called as substrate level phosphorylation reactions. Anyway, here the succinyl CoA synthetase, or succinate synthetase, you can call it as. So converts this succinyl CoA into succinic acid or succinate. Okay, so by removing this thionyl CoA, this is called thionyl CoA. Thionyl means sulfhydryl group unte danni thionyl antam. So this thionyl CoA is removed from the succinyl CoA by this succinyl CoA synthetase or succinate synthetase. అండ్ ఇక్కడ మర్చిపోయాను, ఇక్కడ ఐసోసిట్రేట్ నుంచి ఆల్ఫా కీటో గ్లూటరేట్ కన్వర్షన్ లో ఒక NADH ఫామ్ అవుతుంది, అది గుర్తుంచుకోండి. అండ్ సేమ్ వే ఆల్ఫా కీటో గ్లూటరేట్ నుంచి సక్సినల్ కోఏ ఇక్కడ డిహైడ్రోజనేజ్ అనే వచ్చింది అంటే అక్కడ ఒక NADH2 అనేది ఫామ్ అవుతుంది. ఎలక్ట్రాన్స్ ఆర్ రిసీవ్డ్ ఫ్రమ్ ది ప్రీవియస్ కాంపౌండ్ అని అర్థం. సో ఇక్కడ వరుసగా రెండు NADH2లు ఫామ్ అవుతాయి. ఒకటి ఐసోసిట్రేట్ నుంచి ఆల్ఫా కీటో గ్లూటరేట్ ఫామ్ అయ్యేటప్పుడు, అండ్ ఆల్ఫా కీటో గ్లూటరేట్ నుంచి సక్సినల్ కోఏ ఫార్మేషన్ అప్పుడు వరుసగా రెండు NADHలు ఫామ్ అవుతాయి, అది గుర్తుంచుకోండి. రైట్. అండ్ ఇమీడియట్లీ నెక్స్ట్ రియాక్షన్ లో GTP అనేది ఫామ్ అవుతుంది, దట్ ఈస్ లేటర్ కన్వర్టెడ్ టు ATP ఫ్రమ్ సింగిల్ ఎస్టెల్ కోఏ, గుర్తుంచుకోవాలి. ఇప్పుడు రెండు సైకిల్స్ జరుగుతున్నాయని మనం ఇమాజిన్ చేసుకోవాలి. రైట్. సో హియర్ thionyl CoA is removed, GTP is formed. So to produce succinate, okay, succinate synthase enzyme అనేది దీన్ని catalyze చేస్తుంది రియాక్షన్. రైట్. సో దిస్ succinic acid, okay, is converted into fumarate immediately. And here also two electrons are released from this, two electrons and two protons are released from this succinic acid. Okay, SDH, okay, succinate dehydrogenase. SDH is very, very famous enzyme. Okay, it is attached to actually inner membrane complex. This SDH గుర్తుంచుకోండి, మళ్ళీ వస్తుంది ఎలక్ట్రాన్ ట్రాన్స్పోర్ట్ చైన్ లో. సో SDH is a membrane bound enzyme that always converts this succinate to fumarate. Okay, here the released electrons and protons are added to FAD to convert this into FADH. So that is next fumarate is found from this succinate by liberation of FADH2. Okay, then this fumaric acid, by addition of a single hydrogen water molecule by the enzyme fumarase, is converted into malic acid. Simple, the double bond is removed and water molecule is added, so one hydroxyl group is added. So that is malate. And the ultimate reaction where the another final NADH2 is also produced in this reaction is, so the formation of oxaloacetate in the presence of malate dehydrogenase. Here it is removal of hydrogen takes place, right? So that is and electrons are also removed from this compound, that is given to NAD plus to form another NADH. Okay, overall 3 NADH2 are produced, one FADH2 is produced, okay, one GTP is produced. Anyway, what is formed here from malate to oxaloacetate is formed. Okay, so which is a keto compound, and again it is it re-enters into Krebs cycle, okay, by combining with the acetyl coenzyme A. So with this, the citric acid cycle is occurred in the matrix of the the mitochondria. Okay, in this TCA cycle, 3 NADH2 are produced, one GTP is produced, one FADH2 is produced for one acetyl CoA or for one pyruvate. Okay, రెండు pyruvate ఉంటాయి కాబట్టి మనం ఏమనుకోవాలంటే ఒక గ్లూకోజ్ నుంచి with Krebs cycle, so 6 NADH2 are produced, okay, and 2 GTPs are produced and 2 FADH2 are produced అనేది మనం అండర్స్టాండ్ చేసుకోవాలి. సో దిస్ ఈస్ సమ్ ఇంట్రొడక్షన్. ఓకే, దీని ఫుల్ స్టెప్ బై స్టెప్ నెక్స్ట్ ఈవినింగ్ క్లాస్ లో వి విల్ సి. 7:30కి యాజ్ యూజువల్ గా ఉంటుందన్న క్లాస్, రైట్? సో లెట్స్ డిస్కస్ అబౌట్ ది Krebs cycle and the electron transport also, if possible, time permits. Okay, right. So now I'm stopping this,